What do you think? Just walk me through the logic of what you think is going to happen in this experiment. Anybody? Sorry, let me just share my screen. Yes, go ahead. Go ahead.
The lag phase will decrease.
No, which case are we talking about?
The initial one from time 0 to A.
Why is that?
Because the number of cells will be different in different samples. So the samples with less number of cells will take more time to grow. But a sample taken from C will already have more number of cells, so it will grow faster than others.
I didn't quite understand what you're saying. Say that again.
We are taking same sample size, but each sample size has different number of cells in them, right?
No, so the way the experiment is going is that this point is n naught here, and you start off with n naught cells, and you let them grow to, in this case, na number of cells, and out of those na you pick n naught again, right? So you take n naught out of this na and put them in a fresh tube or a flask and let them grow again. And as soon as they reach Na, you again take n naught out of them and do this again. In this particular case, you let them grow to Nb, and then out of these Nb, you still only take n naught out and put them in a fresh flask and let them go to Nb again, and you again only take n naught out of them. So you're only taking for each of these three situations, you're starting the experiment with only n naught cells. Does that make sense?
Yes, sir.
Yeah. So anybody? Yeah, go ahead. Go ahead, Rishi.
The graph should be the same for all the cases
Okay. Why do you say that?
because everything remains the same. The time taken for the cells to divide, the number of cells in the initial time,
Mm-hmm.
all the conditions are same.
Mm-hmm. Okay. Others?
So I think for A it will remain the same, but for B we will see that the lag phase is much smaller because most of the cells are already in the log phase of growth. They will retain this growth even in subculture.
So yeah, I understand what you're saying. But the question is slightly different actually. Let's say that these cells are being grown in a particular environment. So this experiment is being done where galactose is the sugar, right? And in galactose, we are witnessing something like this. The experiment is as follows: that you do this, they go up to Na, right? And expanding from N0 to Na maybe is 3 generations. Expanding from N0 to Nb is maybe 6 generations, and N0 to Nc maybe is 10 generations. So you grow them for these 3 generations, then you take N0 out of them randomly, and you subculture them in a fresh galactose tube, and you grow them again for 3 generations, and so on and so forth. So you keep doing this growth in galactose till you reach 1000 generations. After you reach 1000 generations, for each of the 3 cases, and the number of transfers for each one will be different, because first one, it's only 3 generations per culture. So you're going to have to do some 333 or whatever number of transfers. So the number of transfers for each one of them is going to be different. But at the end of it, you have these 3 cultures, each of which has been through 3 generations. 3 cultures, each of which has been through 3 generations. 3 cultures, each of which has been through 3 generations. 3 cultures, each of which has been through 3 generations. 3 cultures, each of which has been through 3 generations. 3 cultures, each of which has been through 3 generations. 3 cultures, each of which has been through 3 generations. 3 cultures, each of which has been through 3 generations. 3 cultures, each of which has been through 3 generations. 3 cultures, each of which has been through 3 generations. 3 cultures, each of which has been through 3 generations. 3 cultures, each of which has been through 3 generations. 3 cultures, each of which has been through 3 generations. 3 cultures, each of which has been through 3 generations. 3 cultures, each of which has been through 3 gen 1,000 generations in a galactose environment. Now what you can do is one of the main benefits of working with microorganisms when you're doing an evolutionary experiment is that when you are doing this experiment at 1,000 generations, suppose you want to take a vacation or something, right? Because you've been coming every day and for the last 333 days, you have not taken a break. So what you can do is you can take these cells after 1,000 generations and you can store them in a freezer at minus 80 degrees Celsius. Right? And there is a certain way to do it, and you have to add a few things to make sure the cells don't freeze to death. But what you do is basically take these cells and you freeze them at minus 80. And the benefit of doing that is that you can choose to come back whenever you want, and the cells will be in the exact same state as you left them. So you can just take your culture out from the freezer and see what and do the characterization or whatever needs to be done with them. Right. Another benefit of this is, suppose you wanted to do this experiment for 2,000 generations. Now what you will do is, typically you when you are doing this experiment, after every few hundred generations or something, you will make a freezer stock, this minus 80 degrees Celsius freezer stock every 200 generations or so. So one of the things that's likely to happen, I mean, there is a chance that it will happen, is that you will have contamination someday. Now, if you are at generation 850 and this contamination happens, if you had not paid the freezer stock every 200 generations, what that means is that you have to start the entire experiment from all over again from generation 0, and then that's like 250 days. But if you're making freezer stocks along the way, then you only have to go back to generation 600, which you have already gotten stored, and then continue the experiment from there. Right? So in the context of what we are discussing, what has happened is that through these 3 types of propagations of 1,000 generation in galactose at size Na, Nb, Nc, after this 1,000 generation in each of these 3 cases, you've made these freezer stocks, and now you come back and you revive them. You basically start these cultures again in galactose with N0 number of cells, and you want to see what has been what is the growth trajectory in each of these 3 cases, in each of these 3 cases as a function of time. What does the growth trajectory look like? Right. So what you are saying does make sense in terms of daily transfer, but we are saying after everything has been completed, what does the analysis look like? Does that make sense, Yash? All right, other thoughts?
So, like you had said last time, that in the later phases when glucose concentration would reduce, like the nutrient concentration, so they might adapt to using something like acetate or some other nutrient. So probably in C, there would be a good amount of population which would adapt to using another nutrient. And then at the end of it, when we grow all 3 of them in glucose, probably in C, it'll take more time to grow because it is adapted to using another nutrient like acetate.
Yeah, exactly. Yeah, good. That's pretty good. Yeah. What do others have to say about this? That C might actually be the worst because of this reason.
But on the other hand, it will be the best to perform in the same environment.
I'm sorry, say that again.
On the other hand, it will be the best to perform in the same given environment. If it is continued in galactose, C will be the best. It will be adapted to the size. I mean, the selection pressure is, they have to be of certain size, and energy consumption also be optimal.
But why do you say C is the best adapted?
Because when overpopulation is faced, they will adapt themselves for efficiently using the nutrients available.
But when that overpopulation scenario comes, well, I know I said glucose last time and I said galactose this time, but let's say it's glucose. But when that overcrowding scenario comes into picture and this competition kicks in, there is no glucose left. So whatever adaptation has to happen in that nutrient starved environment has to happen when all the glucose is gone. Does that make sense? Yes.
Yeah.
So how could that competition in an environment where there is no glucose lead to adaptation to glucose? Right?
But it's not like all the, I mean, yeah, eventually all the glucose will be consumed. But the cells which had survived for that long will be those which have the least glucose requirement, I guess.
Okay. Well, okay, sure. That's the thought. Sure. Others? Somebody else?
Sir?
Yes.
Yeah, so I think in the third case, the log phase will last longer, but then its slope will be smaller than the other cases.
You mean the lag phase will last longer?
No, the exponential growth phase.
Will last longer.
Yeah,
Why do
because
you say that?
it can utilize acetate and other things in the medium.
Okay. Okay. So you're saying that growth will happen for a longer time in the third case.
Yeah, but then since the number of active cells in the initial inoculum was small, the slope will be smaller than the other cases.
But the initial number of cells in each of the three cases is the same, right? It's a not.
No, the number of active cells. So from the third, when you take something from the stationary phase, it's possible that there are a few dead cells as well, right?
I understand. Good. Good. Yeah, that's. I mean, something like that is definitely possible. Good. Any other thoughts?
Well, in case B, I think the stationary phase and the first lag phase, I think they'll be shorter. I mean, the lag phase would be slightly longer, and the stationary phase would be shorter, but the exponential phase would be longer.
In B.
In B, yeah, when we take it from the log phase.
Why do you say that?
Because the way I see it is because they've been adapted to B. To the log phase, then it takes more time for them to go to the log phase.
Why does it take longer for them to get to log phase?
Because they might not be well adapted to the lag phase, because we keep taking them off of. I know it doesn't really make sense.
No, it doesn't matter. You go ahead.
It's just that. So if we keep taking them from the log phase,
right,
we expect that they're already, and it's the same environment through and through, right? Every fresh start, so they don't need that much time. Oh yeah, so that would go against my case, because
that makes the lag shorter.
Shorter, yeah. So yeah, shorter log, shorter stationary, and a longer exponential.
Why do you? What does shorter stationary even mean?
Before it reaches death phase, before everything declines.
So why do you say shorter? You mean to say they start dying sooner?
Yeah, very quickly. Yeah.
Why do you say that?
Because because you keep moving them from the exponential. They never. Because we take cells from the log phase, they haven't had time to adapt to the stationary phase.
Yeah.
So most of them will just simply die as soon as the stationary phase comes. Not as soon as it, but it wouldn't be longer than the ancestor.
I understand. That makes sense. But the same comment as what I said to Yash about you commenting that the lag phase will be shorter. What you're talking about is the serial propagation, that when I take them out of log phase and I transfer them to a fresh tube, they've already got all the enzymes made. So the classical the preparatory phase doesn't need to take place. But we are not talking about kinetics after every transfer. We are talking of kinetics at the end of 1000 generations, when you have stored them in another media, and then now you reintroduce them in the media in which you did the evolutionary experiment. Does that make sense?
Yeah, it does.
Yeah. Okay, cool. All right. Okay, sure. So we have a few opinions. I want somebody to comment on case A. What is happening in case? What do you anticipate will happen in case A? Anybody? If you have spoken, not spoken, doesn't matter. But tell me what you think. I mean, to my mind, I think that's sort of the most predictable and also the most interesting, and also perhaps it's the most predictable and perhaps the most non-intuitive case, and it's also the simplest case. And Sangamitra, before you start, and it's also something I didn't have chance to go through all of them, all of your submissions, primarily because I received them at 4 and 9:30 and so on. But I haven't had chance to go through all. But I think case A is something that nobody got right. Yeah, Sangamitra, go ahead.
I thought it might be like the cells will be dead anyway, like because they weren't replicative. So even though we are subculturing, it won't like increase in numbers. So it will be parallel to the time.
But they are increasing with number, right? Because you start with N0, and the whole premise of case A is that you let them grow till NA, and it's just that you don't let them grow for very long, N0 to NA, 3 generations. And then out of that, you again take NA and start all over again. So there's definitely growth happening. I mean, you don't care for time, right? It's maybe not. Just 1 second. It's not as if that you are doing the subculture every 3 hours and that 3-hour period is fixed. No, you wait till that number reaches NA, and that wait could be 3 hours, that could be 30 hours. It doesn't matter. You wait till the number reaches NA, and then you subculture and not. Go ahead, Sangamitra. Anirudh, I'll come to you.
Then maybe the lag phase will be like very high, and the slope will be the least in A. And with time, with B and C, the slope gets steeper, and the lag phase gets shorter. That's how I think it works.
Okay, Anirudh.
In my in the image I sent to you, I actually mentioned that the lag phase would be much longer, but now that I think about it, I think it's shorter. It gets shorter because we don't allow the cells to go through the next 2 phases. For example, most of the time it's within that limit.
Yes.
In the lag
Not
phase.
most of the time. Every time.
Every time, right? Every time.
Yeah.
So it gets adapted to this phase.
Yeah.
And therefore the cell growth becomes better and better, and therefore the lag phase reduces much quicker over time, and it reaches, right? Okay.
Absolutely. Yep. So see, that's weird, right? Because I think the A case was the simplest, but also something that we don't think about. It's simplest because in A case, there is only one selection pressure. The growth phase isn't very long, right? So it's not as if that you're going through a very large expansion. The N0 to NA expansion is really small. So even if you picked up a beneficial mutation and you had a 2% growth advantage, that's not really a great advantage in the case of A, because you never get time to exhibit that advantage. In 3 generations, if you have 1% advantage, it's going to take you several propagations to show up even a minute advantage in terms of increase in frequency, because you're not letting it propagate and you only have 1%, 2% advantage. What is limiting in case A is the fact that you have to wait 5 hours before that small expansion even starts, right? So if you are an individual which picked up a mutation that will grow 1% faster, that you still have to wait 5 hours, but then you grow 1% faster, it's going to be a very, very small advantage. On the other hand, if you are an individual that could shorten the lag from 5 hours to maybe 4.5 hours, that is a huge advantage of half an hour that you've got over everybody else. Because I would say even if that advantage comes at a cost of reducing your growth rate by 2%. So in the exponential phase, even if the reduction in lag phase comes at a cost of reducing your growth rate from 1 to 0.98, that's still a huge advantage, because that 0.02 disadvantage is not going to hurt you very much, because the exponential phase there isn't any. But this half an hour advantage that you got allows you to expand while the others are still in their preparatory phase. So the entire selection pressure in case A is for an individual. I am going to pick up those mutations which shorten lag. Does that make sense to everybody? Yes, no?
Yeah, I agree.
So, as the number of mutations means the chance is very less of any beneficial mutation. Won't the lag phase become constant after some time? Like
No,
gets a
no.
small lag, right?
Say that
And it
again.
becomes
I'm
constant.
sorry. Say
As
that
the
again.
number of beneficial mutations arising in this lag phase, I mean in the A phase, is very less, and we are going till 1,000 generations. Like it might occur in the very last stage, but till that, won't this period become constant in every experiment? Like after, like a very long number of experiments, because like after reaching A means some n number, we are stopping it, so the speed of growing to n will become constant, like a small time.
I am not 100% sure I understand. Can you say that again, and then let me try and see if I got what you're
Okay, like maybe in the first experiment, if it takes some 2t, and next experiment it takes 1t, and later it becomes small and small till it reaches n. After that, it might become constant, right? Because anyhow,
Absolutely.
the n will be increasing.
Absolutely. Yes.
So that lag period will become a small time, like it will become a constant, right?
No, absolutely. So correct me if I'm paraphrasing you wrongly. What you're saying is that if the entire pressure is on reduction of lag phase, then it is true that I will pick up mutations that make the lag phase shorter, but that will not continue for eternity. There will come a time I might go from lag phase equal to 5 hours to lag phase equal to 1 hour, but I won't go from lag phase equal to 5 hours to lag phase equal to 0. So eventually the beneficial mutations that confer a shorter lag phase are going to start drying up, and there will be no more reduction in the lag phase. Is that what you're saying?
Yes, sir.
Absolutely. So whether 1000 generations is enough for me to reach that stage where lag phase can be reduced, I mean, does the saturation in terms of reduction in lag phase, is that saturation reached in 100 generations or 1000 generations or 10,000 generations, is a matter of context, right? I don't know a priori what will be the situation in this case, and that is something, but qualitatively is what we are saying. Eventually you will reach there, but we don't know when is that eventuality going to come. Does that make sense, Namita?
Yes, sir.
Right. So it's an interesting question, because the paper that we are going to read on Wednesday is a classic example of this. That in 2003, a paper was published where somebody, after evolution for 20,000 generations of E. coli, saw a phenotype emerge. Okay, and that phenotype was so curious. E. coli's phenotype changed so dramatically after 20,000 generations that the author said that this is a speciation event that has happened, that this is no longer E. coli, it has become something else. right? And that experiment took 20,000 generations. Now, in 1983 or 84, somebody else had evolved E. coli in slightly different conditions, and the same change had happened in less than 100 generations. Okay, so while these authors, because it took 20,000 generations, because this change took 20,000 generations, called it a speciation event, but the same change was also accomplished in 100 generations, which is then a trivial observation, right? If it happens so easily, then that's not a great example. So the things are context dependent here. Whether some changes will take place in this case, for instance, in 100 generations or 10,000 generations, that's really context dependent and not so trivial without getting the details. Anirudh?
In sexual beings, for example, we take speciation as the event where two individuals who were from a previous ancestral species no longer mate with each other.
Yeah.
So how would you define that for a bacterial population?
There is no, well, I want to say there is no consensus definition of a species for prokaryotes, for asexually reproducing organisms. But that would be a little unfair, because the definition that you are saying for sexual species is something that was proposed by Ernst Mayr in the 1960s, and it's called the biological species concept. Okay. There are dozens of definitions for species, sexual also. So what you are saying is one of the definitions of species. In asexual organisms, the definition of a species is even more contentious, and that's something that I would rather not get into right now, because last 2 or 3 classes of the course is something that I want to dedicate on just speciation, which is perhaps the most exciting phenotype that you can observe in a species, particularly when you're working with sexually reproducing species. I will add one thing though, that they called it a speciation event because of I mean, typically what you would do is sequence a 16S RNA, and then say what happened. They called it a speciation event because the phenotype that E. coli exhibited, and we look at this on Wednesday, the phenotype that they exhibited is a check for phenotype, particularly in I mean, not so much anymore, because now you go for more sophisticated ways of characterization. But maybe back in the 80s and 90s, this was a phenotype to check whether, if you had an infection, whether that infection was by an E. coli or some other pathogen. So this was sort of a way of characterizing whether it was E. coli species that was the causative agent of an infection or something else. And this transition happened such that now this E. coli is going to show you a phenotype that is not associated with any of the E. coli species, and hence they called it a speciation event. Does that answer that somewhat your question?
So it's like it lost its marker basically.
Well, it gained a property that E. coli is not supposed to have.
Okay.
Yeah. So, I mean, just to give you a little more detail, E. coli started using citrate as a food source. E. coli doesn't use citrate.
Okay.
Okay. So, I mean, I was discussing this result came out from the Pavitra. I'll just come to you in a minute. This result came out from an experiment, from this 75,000 generation experiment that I was telling you about the other day. And this was observed maybe 20 or 25,000 generations into the experiment. So I mean, a few years back, I was chatting with someone from the lab where this experiment was done. And I asked him that, how does one explain this? That you have a phenotype that comes up after 25,000 generations in one example. But if you start with the same organism, that phenotype comes up in 100 generations, right? How do you explain that disparity? And his answer was pretty interesting, and I'll tell you guys once we discuss what the phenotypic change was. Pavitra?
Sir, so we were talking about this mutation which took place, right?
Which mutation?
The mutation in the lag phase, which will
Yes.
decrease the lag phase.
Yes.
So won't the effect of such a mutation be felt more if you take an inoculum from the log phase? Because there'll be more species with that mutation, right?
Yeah. You mean more individuals with that?
Yeah.
Yeah. Sure. No, absolutely. No, the point is not comparing it with B. The point is that in scenario A, the only selection pressure is to reduce lag phase.
Okay.
Right?
Okay.
Because increasing growth rate doesn't help you. Right? Surviving in stationary phase, which would be a requirement in case C, doesn't help you because you never get to stationary phase. The only thing that's beneficial in scenario A is reducing lag phase.
Okay, yeah.
Right? That's the only point. So in the graph that you draw, for case A, the only thing you can say with certainty is that A will have a shorter lag phase. We don't know whether that shortening of lag phase comes at a cost of reduction in growth rate, or it increases growth rate. The manifestation could be anything, it could be nothing. The growth rate could remain the same. But the only thing we know is that the lag phase. Sangamitra, what do you have to say to that?
Yeah, it does make sense.
No. If you are still uncomfortable with it, tell me. I don't mind spending more time on this.
No, sir, I'm kind of convinced.
Yeah. Okay. Cool. Fine. So fine. So now let's pick up that same logic. What happens in case B? Somebody tell me.
A shorter lag phase and sharp log phase, I guess.
A short or a sharp log phase? What did you say?
No, I mean a short lag phase, similar to A, but also the lag phase will be sharper. Slope will be higher.
Yeah, exactly. Right. So now there are two selection pressures in case B. That anything that shortens lag phase is a beneficial mutation, right? But anything that increases growth rate is also a beneficial mutation. Increase in growth rate was not really a beneficial mutation in case A, because that was essentially a neutral mutation, right? So now you have two things which are beneficial, and hence case B becomes harder to predict, because because these two beneficial mutations are going to compete against each other in the culture, and that becomes a case of clonal interference that we've been discussing. These two beneficial, and we don't know which one will win in a particular scenario, and so on and so forth. But essentially, if I was drawing the graph for case B, I would draw a shorter lag phase and a faster growth rate, exactly what Yash said, right? I would imagine that in case A, the lag phase gets reduced by a greater amount as compared to case B, because case B, some of the beneficial mutations are going to increase the growth rate, and some of them are going to reduce the lag phase, assuming independence. But in case A, every beneficial mutation has to be of the nature which reduces lag phase. Case C. Somebody.
Will the maximum capacity of the culture increase?
Why do you say that?
Because only those are surviving which can handle the selection pressure well, right? Overpopulation plus the nutrients are going.
But why should the carrying capacity of the environment increase?
Only those variants are surviving which can handle these kind of harsh pressures.
Right, right. But what happens to the lag phase? So let's discuss this. So you reach stationary phase, right? And now some will die because you're spending time in the stationary phase, and only a fraction will survive, and the survival will be contingent on you being able to survive in that stationary phase. But what happens to, let's first answer this. What happens to the lag phase and the log phase?
Shorter and steeper.
Okay, why do you say that? Same logic as B.
Yeah.
Okay. Anybody else?
I think the selection pressure in stationary phase will overtake these advantages.
Sangamitra, what do you say to that?
I couldn't hear what?
So what Yash is saying is that yeah, Yash, I can paraphrase what you say. What he's saying is that there are two things happening. One is that shorter lag phase and higher growth rate are obviously going to be beneficial, but there is another sort of a beneficial mutation that's in case C but not in case B. And that beneficial mutation is that once you are starved for nutrients, once you enter stationary phase, you need to be able to survive in order to make it to the next culture. If you are a genotype which grows really fast, right, but cannot survive in the stationary phase, in case C, that is of absolutely no use because you're going to die as soon as you are starved of nutrients. So in order for a particular genotype to get propagated in the next flask in case C, the essential quality is that you need to be able to survive stationary phase. So what he's saying is that the selection pressure is not therefore on growth phase, growth rate and lag phase. The selection pressure is on survival in stationary phase. What do you say to that? Sangamitra.
I think it will just add to it.
So what you are saying is that you need not worry about it because anybody who is growing faster can also learn to pick up a mutation which increases survival
Like
in
the
stationary
best
phase.
of all, yeah, like it will add up for sure though.
Agree. Sure. I understand. Yeah. So I may have told you. Do you guys remember when the first evolution experiment with bacteria was done? No one remembers. Guess.
30s?
1930s?
Yeah.
Okay. Others before, after that?
But before that, no way.
Yeah. Huh?
But the LB, I mean, it kind of depends on when the media was made, right, for culture.
Yeah. So the first evolution experiment with bacteria was done in 1880s. We briefly talked about this in one of the earliest classes, the course, where it was done by this guy called William Dallinger. And after having read Darwin's book, he wrote to Darwin asking that the principles of natural selection that he talked about in his book, would they hold consistent for microorganisms? And Darwin wrote back that he didn't see any reason why those principles would not hold for a bacterial culture. So he started this experiment with a microbial species. We don't really know what was that species. And the species would only grow, and I'm making up some of the numbers here. So the species would only grow at a temperature around 30 degrees Celsius or something. And he continuously grew it in an environment and slowly jacked up the temperature of the culture media. And this experiment was done from, again, I might be off by a few years, but something like 1880 to 1886. The experiment went on for 6-7 years. Again, we don't know how many generations, because those things weren't characterized back then. And at the end of this whole thing, that species was growing at a temperature around 70 degrees Celsius. So there was a huge increase in the temperature in which this species was surviving. But Dallinger did something interesting. At the end of the experiment, well, the experiment ended abruptly because his experimental setup just blew up. But before that happened, he tested one thing. He took the ancestral strain, and at the start of the experiment, he had grown that ancestral strain at different temperatures. So when he grew this species at 30, it would grow, but he also grew it at 40, 50, 60, 70, and so on and so forth. And at 70, the species obviously didn't grow at all, right? So there was this property that you can either grow at 30 or grow at 70, but not both. And this was a property in the ancestral strain. After having been evolved for a few years, before his experiment ended, what Dallinger did was took this evolved strain, which was growing at 70, he grew it at 30 and compared the growth with the ancestral strain. And what he noted was that having evolved to grow at 70 came at a cost of not being able to grow as well as the ancestor at 30. Does the experiment make sense?
Yes, sir.
Right? This was the sort of the first ever case of what is today called trade-offs. That when you are evolving organisms in a particular environment, you cannot have best of everything. If you evolve to be better in one aspect of physiology, it will have some repercussions in terms of you not being able to do as well as the ancestor in another part of physiology. Now, what that other part of physiology might be is context dependent, and perhaps there is a setup where there is no other cost associated with it, or we don't know of that cost yet. But what Dallinger demonstrated for the first time was this whole phenomena that trade-offs exist, right? And it's the same here. If I had to guess about part C is entering stationary phase. Stationary phase bacteria are going to be ones which are metabolically not very active. They're just there. They're sort of metabolically dormant, and they know they can revive their metabolism whenever there are nutrients. But for the most part, they just linger around and can deal with the nutritional stress for a long time. Now, I don't know the exact trade-off relationships between a dormant metabolism and being able to grow at a high rate, but I would imagine that if you take these stationary phase specialists and compare them for their growth rate will be lower as compared to those mutants which you have isolated from experiment done in case B. In case B, you are selecting for higher growth rates in addition to shorter log phase. But if you were to compare the growth rates of mutants that you have isolated in C and B, it's almost certain that the mutants you isolate in C will not only have growth rates which are lower than the mutants isolated in B, but also lower than the ancestral wild type, because you are selecting for stationary phase survival, which often comes at a cost of reduced growth phase of the species. Okay, so that is the trade-off, that you can't be good at stationary phase survival and exhibit high growth rates. There is a trade-off that you're selecting against. So in terms of our experiment, case A is the easiest to predict because there is only one-dimensional selection pressure on it. Case B is the next harder bit to predict because there are two selection pressures. Shorter lag phase is also beneficial. Higher growth rate is also beneficial. And case C is the hardest to predict because now we have entering stationary phase, and stationary phase physiology is very, very complex. That's why most of the people, when you do experiments like this, when you're doing evolution experiments, remember there are three ways of doing evolution experiments. We are still on the first one. When you're doing something like this, what was I going to say? Yeah. So when you're doing, so typically any paper you read, you will see that the authors make sure that whenever the transfer from one flask to other takes place, it takes place before the cells get into stationary phase. So stationary phase physiology is very, very different from actively growing physiology of the cell. And unless you are studying stationary phase exclusively, if that is your core area of expertise, you don't really want the cells to get into stationary phase. You just keep them in the vegetatively active growing phase, and you just keep propagating before they hit the stationary phase, because that's something that's not very well understood at all. All right, does that make sense? Yeah. All right. So a couple of comments. Again, we discussed this when we were discussing the Illinois crop experiment earlier in the course, but very important to remember that when you are doing an evolution experiment with a microbial population, right, you can process generations very fast. When you're working with bacteria or yeast, typically you would do a 1:100 dilution every 24 hours or every 48 hours at the latest. 1:100 dilution comes to about 6 to 7 generations. So in a week, you can easily do 20 to 50 generations depending on how fast you subculture. So you can process generations very fast. And if you are processing generations very fast, what that means is that you're picking up beneficial mutations also very fast. Population sizes are large, and hence that means you are sampling a lot of mutations every day, and then selection is acting on those mutations and so on and so forth, right? So that's a feature of evolution. That's a core feature of how evolution experiments are designed when you are working with bacteria or yeast. If you are working with another model system for experimental evolution, let's say Drosophila, right? Who does not know what is Drosophila? Just be shameless, just raise your hands. Everybody knows? All right, cool. So if you're working with something like a Drosophila, now you're dealing with a fly. So having you can't have populations as large as the ones you were working with when you were working with bacteria or yeast. So population size becomes extremely small. The other issue is that generation time is now much longer, right? With Drosophila, it will be of the order of weeks now. So that's severe. So population size is small. Hence, what that means is that you're going to have to wait a long time for beneficial mutations to arise. And second is that now the generation time has gone from a few hours to of the order of weeks. What that means is that you have to wait an inordinately long time for beneficial mutations to arise. So how would you study? But there are lots of Drosophila, lots of labs which address questions of evolution working with Drosophila. How do they do that? We discussed this. So when you are working with a population such as Drosophila, you are not relying on new beneficial mutations to arise, and hence natural selection act on that act on this new genetic diversity that has come via mutations. That is not going to work out because of the timescales involved. What you are instead relying on is meiosis to create genetic diversity. That I'm carrying a certain alleles, my partner is carrying certain alleles, and if we mate, then just meiosis and recombination is going to mean that the offspring has genetic diversity, right? Now, obviously, this genetic diversity will only be generated if me and my partner are non-identical genotypes, right? If we carry different alleles, because if our genomes are identical, then meiosis and recombination is not going to generate diversity. But so in sexually reproducing, I mean, yeast is also sexually reproducing, but in multicellular exclusively sexually reproducing populations like Drosophila, evolution experiment is done with the aim of studying where the focus becomes not the new beneficial mutations that arise, because that's going to take a very, very long time. The focus becomes how does the existing genetic diversity in the population that I'm starting with change in a particular environment. Suppose I'm adapting Drosophila to 50 degrees Celsius in highly humid conditions, and I'm starting with a population which carries different alleles at different loci. Now, any allele that gives you advantage in terms of survival and reproduction at 50 degrees Celsius is going to increase in frequency as I go forward in generations, because that allele is beneficial in nature and hence is going to lead to more offspring. So the next generation, that allele is going to be overrepresented, and so on and so forth. The population will progress then. So in a population such as that, you are not looking at new alleles to arise. Well, not via mutation anyway. You are looking at how does the frequency of existing alleles change with time. So there'll be some alleles which are really good for survival and reproduction at low temperatures. But this 50 degrees Celsius is going to weed them out, because again, there'll be a trade-off involved. You can be good at 10 degrees Celsius or you can be good at 50, not both. So these alleles will decrease in frequency as the experiment progresses, and those which help confer a higher fitness. Fitness, remember, for Drosophila would be number of offsprings you leave behind. Those alleles which confer a greater fitness at 50 degrees Celsius are going to increase in frequency. So what you are studying in a scenario like this is that as you propagate this population for, typically you would do a few dozen generations. When you do this few dozen generations, what did the allele frequency change take place in this environmental context in which you did the experiment? So fundamentally different way of how you are studying evolution in a bacterial and a yeast population compared to what you're doing in a population of, let's say, flies. Does that make sense?
Yeah, I have a question here though.
Yeah.
So if just curious, at the time of the theory of dominance, Fisher's theory of dominance, he didn't take into account the process of recombination, because based on his idea, it was basically always a mutation of the modifier genes, for example.
Yeah.
Right? So would that be basically what he's saying is that the process of dominance is completely due to the process of mutation, the rate of mutation?
In the context of dominance, yes, that's what Fisher said.
Right. So recombination does not necessarily have anything to do with dominance per se. It could it. I don't know how exactly, but could it actually affect the dominance of a particular?
Well, it could, but what Fisher is saying is that when a new allele arises, for it to become dominant, recombination won't do it, because this allele did not exist before then, right? And hence you need a new modifier allele which would impart dominance to this particular allele.
But
right?
that couldn't be done by recombination of
It
the
couldn't be.
modifier allele.
It couldn't be, right? Because this new allele didn't exist, and hence the appropriate modifier allele didn't exist. It's only now that this new allele has come that there is a need for, or selection would act on this new modifier allele. But because this didn't exist up until this point, the modifier allele doesn't exist in the populations. Hence, it has to be mutation and not recombination. Right? Cool. All right. Great. So we'll come to the fine. So no more comments. I want to discuss one which I think is a really curious way of doing evolution. Can you think of a way in which we can do an evolution experiment where there is no selection? Anybody? Let me put it another way. How would you measure mutation rates of a species? Can you think of a way mutation rates are measured? And just think aloud. Don't worry about right or wrong. Just think aloud.
This is without sequencing.
With sequencing.
So you could just take the DNA at a varied time span and then check the frequency of changes.
No, say that again. Details.
So, if we can keep a generation gap, like a set of generations, after which you would take one species and check its DNA.
So
sequence its DNA.
Let's sharpen it again. Let's say you have E. coli.
Right. So let's say you want to you'll take the DNA after every 5 generations. So you take a species out. You take an individual out of that, and or a set of individuals, and then you sequence the DNA and you check the difference, the amount of time that generation, those 5 generations took, and then you also check the number of mutations that.
Right. Why does it have to be 5 generations? Why can't I do it after 100 generations?
But in my opinion, at 100 generations, we lose most of the mutations because they're not carried forward. Because we keep changing the meat. For example, we only take N naught, right, at each subculture.
Right.
And if there were mutations, basically our estimate would be much lower than the actual number of mutations.
Right. Yeah. Right. And how does taking samples every 5 generations resolve this?
Well, actually, we should try to do it. We should optimize this experiment. We should try taking it at 1, taking it at 5, taking it at 10, and then see if at two of those generation points, the mutation rate is pretty much the same. So we take the maximum of that. So we take the generation that's at max. Something like that.
Right. Sure. Good. Anybody else? So Anirudh, would it be correct to say that what you are saying is, in order for us to measure a true mutation rate of a species, we have to design an evolution experiment where the effect of selection is negated?
Yeah, right, exactly.
Right, because selection is messing up things. When we have selection acting, what is happening is that all the deleterious variation that has been generated is being weeded out from the population, and I only get to see a few beneficial mutations and perhaps an occasionally neutral mutation. I only get to see variations such as that in the culture after a 100 generations or whatever, right? So selection is acting, and that, remember, mutation is a process that generates diversity. Selection is a process that gets rid of the diversity. And in a bacterial yeast culture, we saw the traveling wave model, right? That there are going to be lots of genetic variation existing within a culture, and selection is acting within this. So we need to come up with a way of doing an evolution experiment where the effect of selection is minimized. How could we do that? Is the premise clear that with selection acting, my estimates of beneficial mutations are going to be, I'm sorry, my estimates of mutation rate are going to be much smaller compared to what the actual mutation rate is. And hence, in order to estimate true mutation rates, I need to come up with an experimental plan where selection is eliminated. How would I do that?
I have a doubt.
Yes.
With Anirudh's plan, where we are very frequently sequencing the DNA,
Yes.
even if selection is in place,
Selection is in place.
even if selection is in place, we will be getting a true mutation rate, right? Because the frequency of sequencing is very large.
So what do you, how do we do this? Let's discuss the logistics of this, right? That you have n naught, and then what typically what you do is you do a 1:100 dilution every day, right? So you have n naught, and you will wait for the population to expand to 100 n naught. So Anirudh, how do we go about this plan?
Where you keep subculturing at a 1:100.
Yeah, we can adjust a 1:100 bit. We can make it whatever you like, at least in theory, right? But my question is that at what frequency do I sequence? How many cells do I sequence?
But the question of how many cells one should sequence is, I think, really it depends on how much have you left behind.
Okay.
So if you leave most of it behind, like you mentioned, 1:100, so that means you kind of leave 100, and you take 1 for every 100.
Yeah.
So so if it were a 1:1000, then I'd expect you take more samples in the next. So at the 5th generation, for example, if we're taking the sequencing, I'd expect we take more number of samples.
Yeah.
And the other way, vice versa, the other way around. But again, it's this problem is also based on optimization.
Yeah, but it's hard, right? Because you're talking of a population size which is of the order of 10 per 9.
Right.
Right.
So I think right. So I think it would. Yeah. It's very difficult that way.
The logistics would be tricky.
I think the ideal way would just you don't subculture at all, but that's practically impossible.
No, so what happens in that scenario?
So if you were, if that's the case, you just keep taking samples at a time interval, and so even in the same time, you take different samples. For example, take 100 samples at time t or at generation n, and then you can, because it would act like a random variable. We have to make that assumption first. We can plot a Gaussian distribution of the values for each generation, and then every next generation, or maybe every 3 generations, if you calculate the plot, that mean is also a random variable, and you again make a distribution, and you eventually find out per generation, this is the so something like that. I think that makes sense.
Yeah. Well, so typically when you do that, That also sounds tricky to me, because what you're going to do is when you sample at a particular point, you're going to get genetic diversity in your sample, right? And when you get your sequencing results, so at t equal to t naught, you took 5 samples, right? And you sent one of the samples for sequencing. At t equal to, for this sample, when the sequencing results come, what you're going to get is two things. Suppose half the mutants in this one carried at position 100A, and half of them carried a G at nucleotide position 100. So that you would see, right, that A and let's say C or a G, whatever, right? A and a G at position 100. But for mutants which are present at very low frequency, you're not going to see them, right? So all of the genetic diversity which is actually deleterious in nature will be pretty hard to detect even in the scheme. Does that make sense?
Right. So as we take more samples in the same generation, I think we're more likely to come across that mutation. But like you said, I don't think we can cover every mutation that is there.
Not just that. I think it's more serious than that, because the nature of deleterious mutations is that there will be a lot many of them, but they will not go up to very high frequency. And when you are sampling for sequencing, it's the frequency that matters, right? Essentially, when you are sending a sample for sequencing, what you are asking is that in this collection of cells, what is the frequency of nucleotides at every position? And the nature of deleterious mutations is going to be that there are many of them, but none of them is going to be at a very high frequency, and that becomes the problem. Does that make sense?
Yeah. But maybe instead you could just use an in vitro system where you use the DNA polymerase of that particular bacteria, and you have a particular DNA construct, and you keep doing this experiment and you keep sequencing the DNA until you find one or two mutations, and then those mutations don't die away.
Yeah, sure.
So in that scenario, we can accurately calculate the amount of mutation and the mutation rate.
Yeah. So that's interesting. What is done is an in vivo version of that. Exactly what you said. Okay. How do we do that? And that is sort of the third way of doing. For the time being, I'm skipping the second way of doing it. That's the third way of doing an evolution experiment. Let me show you what the scheme is. It's called a mutation accumulation experiment. And this is the scheme. That what you do is you take n naught number of cells, and n naught, let's say n naught is 100, right? You take a fresh plate. So you have a little vial in which there are yeast or bacterial cells, and this volume of liquid has 100 cells. If it's 100 cells, you can't see them, but by dilution of a more concentrated culture, you know that there are ballpark 100 cells in this volume. Right? Then what you do is you take a plate and you randomly mark an area like this. I mean, you would literally do this. This is a fresh plate, solid agar plate. You would literally mark an area on this plate like this. Prior to spreading these 100 cells on this plate, you would mark an area. Then what you would do is you would take these 100 cells and spread them randomly. Again, because you couldn't even see the 100 cells in the culture, there is no way you know where you are spreading or where those 100 cells are going. But presumably, all those hundreds are being deposited at different sections of this plate. You have marked an area prior to spreading this. Now what you do is you leave this plate overnight, or if it's yeast, typically 2 days. you leave this for 2 days in the incubator, and wherever you spread these cells, you're going to have colonies now, right? And what you do, so now you have these colonies, and in the area that you marked, so this was the area that you marked, this is the colony that you get. And now what you do is you pick this colony and you suspend this colony, and out of this suspended colony you again take 100 cells, you dilute it to a level so that you have 100 cells, and you plate these 100 cells on a new plate, and you mark an area prior to plating, and so on and so forth, and you keep doing this over and over and over again, right? What is happening in this experiment? That because you mark an area prior to spreading, you are saying that of these 100 cells, I am going to pick one which lands in this area, and hence that makes your choice completely random, no selection, right? And then when that colony grows, you pick up and you pick one out of all the cells in that colony for the next spread. I mean, you pick 100 and then you spread that 100 and then again you pick. So you are propagating every day, but your propagation doesn't have any selection on it. So what might happen is that one of these days, the guy that you picked has a mutation. It could be any kind, but because there is no selection, because you mark that area prior to spreading the cells or prior to seeing the colonies actually develop, you are forced to pick that particular colony, and this will have a mutation. And you keep doing this over and over again, and eventually the individual, so now on some day this guy had a mutation, so I am picking an individual which had a mutation, because the cell that got spread here, when I was doing the spreading, the cell that got plated here had a mutation, and hence everyone in this colony is going to have a mutation. Now that becomes my founder for the next day. So now this mutation gets picked. Now by just random chance, if I do this for long enough, I am going to pick up another cell which had a mutation now. So that becomes now my cells which are being plated have 2 mutations now. And gradually, as I keep doing this over and over again, the number of mutations that keep getting accumulated in this, in what is called a MA line, mutation accumulation line, is going to get increased in a manner which is roughly linear with time, provided I do this for long enough. And at the end of a very long time, after lots of such propagations, we'll come to what a typical number is. I will sequence that last cell and compare the sequence with the ancestor. And now I know how many generations was it, how many propagations was it, and then I know the exact time between the ancestral and after what I have now, and that gives me the mutation rate associated with the species. Is the logic of that experiment clear?
Yeah.
Questions, comments?
But even here, we are losing lots of mutations, correct?
You're losing lots of mutations, and it's impossible. So essentially what you're saying is that you've taken selection out of the question, because what you're doing is you're selecting a colony at random, right? And who gets plated the next day is also random. So there is no selection acting in this. And because of this reason, the mutations that are getting accumulated in this one cell, remember this one cell has a lineage. This one cell has a parent that started from ancestor. So typically you started with one ancestor which spread into 2, which spread into 4, and so on and so forth. This is spreading continuously. Essentially what you are doing is you are tracing one line from the present day, this individual which has picked 5 mutations, all the way back to the ancestor that you started with. There is a line obviously that connects them, right? And this line has been followed without the action of selection. Does that make sense?
Yeah.
Right? And typically when you do something like this, you are not doing just one line. you are doing several lines in parallel. So you will get a mutation rate estimate from one line, and you are doing this in parallel. So you don't have just one plate continuing, you have many plates continuing, and you will get a mutation rate estimate from each one of these lines. And if you are doing this for long enough, if you're doing this for short enough, one line may pick up a mutation, others might not. But if you do this for a long enough time, then you are estimating a mutation rate from each one of these lines, and they will tend to over out over a longish period of a time. And hence you try to decipher the mutation rate of a species using this particular scheme. Questions, comments?
So if one of the mutations is deleterious and we take that into another culture and start growing it, and it won't grow, right? So would like the line stop there? So you can't do any more experiments after that because it couldn't grow because the mutation just killed that cell.
So
like that.
yeah, so let's take a look. That's an interesting point. So suppose, so what is the scheme here? That I picked this colony, right? This had some n naught amount of, not n naught, this had some p number of cells. Out of these p, see, because these p cells were there, they were alive, right? Now out of these p alive cells, I am picking n naught, right? And because these p cells were alive, because they were there, I have these n naught cells which are also alive. And now I spread these cells, these n naught cells, which is 100, and these cells are also alive, and now they have nutrients, so they will grow. So it is possible that an individual picks up a lethal mutation, but I will never see that lethal mutation, but because it will never grow on my plate. Suppose I picked up an individual which has lost the capacity to replicate, right? The individual is alive, but it cannot replicate. Eventually it will just die of stress. But I will never see that individual, because even if that individual was in my 100, and even if that individual got plated here, because that individual never replicated, I will never see that colony emerge here, right? Does that answer your question, Shri?
Yeah, it does. So, but then in the mutation rate which we get eventually, again, the deleterious mutations aren't we aren't taking those into account at all, right?
You are taking them into account. You are not taking into account lethal mutations. So any mutation that decreases fitness, is going to get picked up, right? That's not an issue. The area that you have marked on the plate, you may have colonies which are growing slower than the ancestor. That's not a problem. You still pick that colony because it's in that marked area, right? So deleterious mutation is not a problem, because that just decreases fitness. What you will never pick up in this assay is a lethal mutation that changes, which is a special sort of a deleterious mutation which makes your fitness equal to 0. Does that make sense?
Yeah, it does.
Yeah. So in fact, when you do a mutation accumulation experiment, and so typically the number of propagations that you do in order to get some sort of an estimate is at least a couple of hundred. Okay. So that immediately means if you're doing this with yeast, for instance, you need 2 days for this yeast to grow. That immediately means 400 days. That's more than a year. Now, what was I going to say? So when you do this for 200 propagations, and remember, it's bacteria and yeast, so I can make freezer stocks. I can make a stock at transfer number 50, I can make a stock at transfer number 100, and so on and so forth. And when my experiment is complete, I have done all 200, I can take these out. I still have the ancestor in my freezer. I have the colony at transfer number 50, 100, 150, 200. And now I can collectively study the growth rates of all these 5. And I can plot a graph where on the x-axis, it's the number of propagations that have taken place, starting from 0, the ancestor, and currently number 200, and on the y-axis I have the fitness associated with it, right? Typically the graph that you get is a straight line decrease. Fitness decreases in mutation accumulation experiments as you move forward in time, and that's not too surprising, because when we discussed earlier in the course that what is our intuition of frequencies of deleterious and relative frequencies of deleterious and beneficial mutations, all of us suggested that all of us anticipate, or our intuition suggests that deleterious mutations are going to be more frequent than beneficial mutations. And as a result of that, in this mutation accumulation experiments, more often than not, you are going to pick up a deleterious mutation rather than a beneficial mutation. And hence, as a result of this, the mean fitness, suppose you are doing 10 different lines, the mean fitness is going to decrease with time as you increase in the number of propagations in this experiment. Does that make sense?
Yes, sir.
Another question?
No, I just said yes.
Yeah. All right. We can go into more details if you have questions. But I just do want to use the last 5 minutes for the experiment that we're going to discuss next week. Yeah. So if you have questions, we'll start Wednesday's class with that. But here is the so now I've deliberately sort of avoided the third way of doing evolution. We'll come to that if it needs us to. But as I said, more than 99% of all evolution experiments are conducted via serial propagation. You do MA experiments for a very, very specific purpose, because normally in an evolution experiment, you are studying the adaptive response of a population in a particular environmental context. You only do an MA experiment when you want to take selection right out of the equation, which you would only do for a very, very specific reason, right? Typically, you wouldn't do an MA experiment. But MA experiments, I think, are curious and interesting because they ask a very different set of questions than a serial propagation experiment, which is answering for an adaptive response of a population in a very precise environmental context. The third one is also a way of asking a question of a population's response to a particular environment. And if time permits, in one of these classes, I'll come to that. But fundamentally, it's not different from the serial propagation. Serial propagation is a lot more easier, and hence people do it that way. All right. So the experiment is as follows. So in 1987, Richard Lenski at university, he was in California then, he subsequently moved to Michigan. So he's currently at Michigan. He started this experiment where E. coli was evolved. E. coli was added to a flask which contained low amounts of glucose. Okay. And the premise of the experiment was that he wanted to study the adaptive response of a population, what sort of mutations, and remember, this sequencing is not happening at this point. So he's just interested in characterizing the growth curve that we started today's class with. In low glucose, so now glucose is starving, so perhaps the kind of beneficial mutations you would pick up is the one which allows you to take up glucose faster, better than your competitors in the population. And in this low glucose environment, he's studying the adaptive response of a population. There are two interesting aspects to this experiment which he did at the time of start. One is, back in the day, a lot of actually microbiological techniques are there only because of historical reasons. So those of you who worked with microbial populations will know that you add yeast extract to your growth media. Show of hands who do know that. All right, a few of you. Yeast extract was only added historically. I mean, this is typically just yeast grown and just ground up to make this powder, and you add this powder to your growth media. Historically, yeast extract was only added because people thought that bacteria and yeast will only grow on sort of animal proteins. They need something from an animal origin, and hence yeast was grown and added to the, and that's the only reason that you add yeast extract. It's an undefined media. We actually don't know what we are adding to the media, but historically that's the reason. So there are lots of such historical accidents which decide protocols today also. Another thing that was done was that people used to add citrate to the growth media. Again, back in the day, early days of microbiology. And the reason for that is that water supply for microbiological experiments wasn't really reliable. There was batch to batch variation. And people saw that if you added citrate to the media, irrespective of whether your bug used citrate as a food or not, if you added citrate to the media, it reduced the batch to batch variation in terms of the growth characteristics of the organism. We don't really need to do that today because we have a greater control over the water quality that we are giving and everything else that we are adding to the media. But still, it's done for several experiments. This was the first thing that, one of the two things that I want to mention about the experiments, because it leads to an interesting outcome. Lenski, to his low glucose environment, added citrate. E. coli doesn't use citrate, so he wasn't particularly worried. This was only done to increase the replication of the growth curves that he saw in his cultures. There is a lot of citrate in the media. The second thing that Lenski did was so now the plan is that I have a flask, there is glucose and citrate in it, and I'm going to subculture it every single day. The second thing that Lenski did was he didn't just start with one flask, he started with 12 such flasks. So I have an identical ancestral population which has been added to these 12 flasks, and these 12 flasks are going to be propagated in parallel. So the starting point on the landscape for these populations is the same, and from there they are adapting to an identical environment, and I'm going to study what is going to be the evolutionary fate of these 12 populations. And since that time, I'm guessing close to 100 papers have been published on this experiment. And there was a time around 2000 when Lenski says that nothing really interesting is happening in this experiment, so perhaps it's time to close this experiment. And he was at around 20,000 generations, but he still continued for a bit. And then something interesting happened. One of the 12 lines started using citrate, right? So now in one line, we have this citrate user. And how would you detect it? You detect it because when you come for subculturing every day, you are accustomed to there being a certain amount of density of the culture because you do this every day, But in the line that started using citrate, when you come in the morning, you note that the density is considerably higher because you have so much more growth, because citrate has also been used up. You have so much more growth than compared to every other flask, and based on your experience. Now, your immediate thought when that happens is that there is contamination in the media. It wasn't contamination. In this line, E. coli learned how to use citrate. And we're going to study, and this is an example of a metabolic innovation. And we are going to study that what sort of genetic changes would have brought about this phenotypic change. The second question that we'll study, which is really interesting, is because this is an experiment which has been going on for such a long time now, 75,000 generations, and Lenski is now retiring, so this experiment is moving to one of his former students who is now a faculty at Texas Austin. So the experiment is going to move there. Now, the second question is that is there a fitness peak, right? Or are these lines moving to different peaks? We can answer this question phenotypically by just monitoring the trajectory of growth rate with generations, that as generations are moving, is the growth rate saturating or has it saturated, and so on and so forth. We can also answer this question genotypically by looking at where on the sequence space are the sequences moving to. Are they moving towards different peaks, as Wright would suggest, or are they moving towards the same peak as Fisher would have said? So, as I said, I mean, there are 100 papers on this single experiment alone. I will share two of them, and just come having glanced through these two papers, and we will start our discussion on this on Wednesday. But we will begin our class by discussing if you have questions on MA experiments. All right. If you have quick comments, we can do that now. Otherwise, let's stop for today. We'll pick this up on Wednesday. Feel free to leave if you don't have any questions.